Nyalimi Productions presents Cell Transport Mechanisms. Have you ever wondered what it must be like to be inside a cell? Imagine the genetic material, the cytoplasm, and the ribosomes. You will find that in almost all cells, prokaryotes and eukaryotes. Eukaryotes cells, in addition, have membrane-bound organelles. All of those organelles have different functions, but cells are not isolated little worlds. It makes sense that to keep a stable environment inside them, otherwise known as keeping homeostasis, they must have some control on what goes in and out of them. A very important structure for this is that all cells contain is the cell membrane. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure and signaling abilities, but to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Part of them is the tail, which is nonpolar, and the head, which is polar. Some molecules have no problem going through the cell membrane and directly go through the phospholipid bilayer. Very small nonpolar molecules fit in this category, and a great example are some gases. Oxygen and carbon dioxide are great examples. This is known as simple diffusion. Simple diffusion doesn't take any energy to force these molecules in or out, so this is known as passive transport. Simple diffusion moves with the flow, meaning out, so this is known as passive transport. It moves with a concentration gradient. Molecules move from high concentration to a low concentration. That's the natural way molecules like to move, from high to low. So when you hear someone saying it's going to be with a gradient, then that is what they mean. Remember how we said that the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some of these proteins actually change their shape to get items across. This is known as facilitated diffusion. It is still diffusion, and it still moves with a concentration gradient of high to low. It does not require any energy, so it is a type of passive transport. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel as fast across the membrane, it passes through protein in channels called aquaporins. Now, all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cell's lining need to take in glucose. But what if the concentration of glucose in the cell is higher than the environment? We still need to get the glucose in, and it's going to have to be forced against the regular gradient flow. Typically, it will require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates. When the bond for the last phosphate is broken, it releases a great amount of energy. ATP can power active transport to force these molecules to go against their concentration gradient, and one way to do that is actually energizing the transport protein itself. One of our favorite examples of active transport is the sodium-potassium pump, so that's definitely worth checking out. There's other times the cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. A big polysaccharide needs the cell membrane to fuse with the molecule it's taking in to bring it inside, and this is called the endocytosis. Think endo for in. Endocytosis is a general term, but there are actual different types of endocytosis. Amoebas, for example, rely on a form of endocytosis. Pseudopods stretch around what they want to engulf and that it gets pulled into a vacuole. There are other forms too, such as the fancy receptor-mediated endocytosis, where cells can be very picky about what's coming in because the incoming substances actually have to bind to receptors to even get in, or pinocytosis, which allows cells to take in fluids. Exocytosis is the reverse direction of endocytosis, so this is when molecules exit. Think exo and exit. Exocytosis can also be used to get rid of cell waste, but it's also really important for getting valuable materials out the cell is made. One cool example? Thinking back to those polysaccharides, did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. All cells have membranes, but not all cells have a wall. But if we're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you need exocytosis right there. So that is it for cell transport mechanisms. I hope you learned something today. Bye!
No, honey, I'm good. I could have another, but I probably should not. I got somebody at home.